Okay. Good morning, everyone. Welcome to our second annual Ladders to Cures Accelerator Symposium. My name is Greka. I'm a core faculty here at the Broad and physician scientist at MGB. It is my great pleasure to open the symposium today. We have a great program lined up, and I'm very excited for the whole day. I hope that you can all, whether you're online or with us in person, you can stay engaged. I just wanted to make some introductory comments about what the Ladders to Cures Accelerator is and where we stand now. This whole activity was launched more than 2 years ago now. Our official inaugural symposium was exactly 2 years ago. But the process, actually, the brainstorming process started maybe 3 years ago when we gathered many of you here in this community to think about how can we take all of our genetic and genomic discoveries that the Broad has been at the center of for so many years and try to harness them for the benefit of patients. And of course, rare genetic diseases are an area that is ripe for translation into therapies, and this was something that we really wanted to see how we can accelerate further. And through basically brainstorming with the entire community, we came up with this idea of launching the Ladders to Cures Accelerator, which is really a scientific accelerator aiming at bridging, genetic discoveries and work in rare diseases, hopefully all the way to therapeutic hypotheses and therapies for the benefit of patients. And so this is really how this idea was born, and it's really been a tremendous couple of years, building this community. This is officially our mission, which was actually very well articulated by our very own director, Todd Golub. He had a lot to do with articulating this mission statement very clearly. Catalyze progress across the research ecosystem and accelerate advances leading to treatments and cures for patients with rare genetic diseases. So this is what we aim to do. It's a big, ambitious goal, but I think the time is right for us to make progress in this space. And so to really try to articulate a little bit further what it is that we're actually hoping to do, the idea is to actually build a platform that can uncover nodal mechanisms across many rare genetic diseases. So I just want to point out that nodes doesn't necessarily only mean biological nodes, although of course that's a lot of what we do and we will be hearing about today, but it can also mean technological nodes. So ways in which we can bring technologies that can be applied across many different diseases and find ways to accelerate getting these technologies to patients. And so, for example, our symposium is timed perfectly with this, incredible story that was in the news yesterday about little baby KJ who, received a base editor to treat his disease at just a few months of age, and he's doing very well, and it's just such a miracle. And I want to celebrate that, and of course the entire rare disease community has been celebrating this news since yesterday, but to also say that that's a technological node, being able to figure out ways in which we can make KJ not the exception that we celebrate, but the beginning of a revolution of many, many other babies or people like KJ who receive these therapies. So harnessing these technologies and finding ways to deploy them for patients in a way that is actually applied, to dozens and then hundreds and maybe thousands of patients, millions of patients ultimately across the planet so that everyone can benefit. I think we will be hearing from Jimmy later today about some of those ideas. But to me, that's a technological node. And a biological node would be, you find a target, like some of the work that, our lab has been working on, and you develop treatments, and then, targeting that particular node actually can de-orphan multiple upstream diseases. That's another way to harness the opportunity to help more patients. But in both cases, I think the idea of finding these nodes, searching for nodes and finding them and then deploying them for therapeutic benefit is really what we're hoping to accomplish. And so, rare diseases are collectively common, and we should find these nodes so we can find therapies that can ultimately help more and more patients. It shouldn't be the exception. It should be something that we do in a highly repeatable fashion as we move forward. That's how I would define success. So as we make small steps in that direction, vision beyond building a platform that can harness nodes, or multiple platforms really for harnessing these nodes. We also are hoping to have some proof of concept therapeutic projects. And so this is what we call our leading edge projects. We have started by seeding 10 of them. You will hear about all of them today. This is part of this symposium, hearing about our leading edge projects and the progress that they have made. It is clearly work in progress because we're only about 6 months into beginning this process of having seeded these projects. So all of the presenters know that this is work in progress that they're showing us, and we're really just all working together as a community to come to progress in that space. Of course, nothing in rare diseases happens without patients. So this is very much a space and an initiative that is led by patients and patient advocacy groups. And many of you will know that in our open call for leading edge projects, leading edge ideas, we actually had a requirement that all of the scientists leading the ideas will actually have a patient advocacy group with them. And we did indeed have that. That was a requirement for the process. And that was really to galvanize this idea that patients are our partners on this journey to therapies, and they're the most important stakeholder for everything that we do. And of course, it goes without saying that all of this requires collaboration. So of course, a big goal of our Ladders to Cures mission is to have a collaborative effort. So just some numbers. So far, we count 29 scientists who are working collaboratively as part of this accelerator. We have 10 cohort projects in our first cohort that you will hear about. We have so far organized 6 symposia and community events. These are our Rare Disease Day events, which we celebrated a decade of those this past February. And we've had, our inaugural symposium and a couple of additional symposia on AAVs, on using AI for the benefit of genetic diseases and making progress, and many other ideas that are part of this important ecosystem so that we can all make progress toward our mission. And finally, none of this would actually be possible without philanthropic support. So I'm incredibly grateful to 2 anonymous philanthropic organizations who support our work, as well as the Chan Zuckerberg Initiative. And so we're just really delighted to have their support and hope, of course, to continue to make progress in that space as well. Okay, I will not read the names of the projects, but I just wanted to show you the breadth and depth of the different projects that we're discussing today. Some of them are leading edge projects that are focused more on nodal biology, and some of them are leading edge projects that are focused more on nodal technologies. But in both directions, I think we're making some good progress in many different diverse areas, and you will see a lot of tremendous ideas being shared by our colleagues later today. This is just to show you the symposia and events that we have organized over the last couple of years. There's been a lot of activity, and of course we hope to continue to bring the community together across many different ideas. And while this was not a priority, we've finally secured some funding to launch a nice website. So we wanted to get into the substance of the science, but now we're also getting a website together so that we have, a sort of a place, a launching pad, if you will, for the accelerator. And so this is coming in the next couple of weeks, and I just wanted to give you a preview that we have our website coming up. And so I just wanted to end my remarks by speaking of the people who are most important for bringing us together today. The first person I want to acknowledge is Dr. Jillian Shaw. No one is luckier than me for having the opportunity to work with someone like Jillian, who really all of you know, I'm sure, because she brings our community together, and she's really the glue that makes all of it happen. This beautiful website and all the logistics for today would not be possible without Katie Liguori. So I'm also incredibly lucky to have Katie in the office, and she really makes everything tick, really, including me. So I'm really, really grateful for everything that they do. But something that you may not know is that Katie actually also has a design degree. So a lot of the designs for what you see, the logos, the website, I'm just so lucky that she actually also puts her, design genius to work. And so we all get to benefit from that beautiful work as well. So very lucky for that. And then just some final logistics for today. We have incredible keynote speakers. I'm about to introduce the first one. But I just wanted to say we'll have Q&A after each of the keynotes. But then when we get into the sessions, and also in the interest of time, we will ask our speakers to sit back down after they've spoken, and then at the end, everybody, if you wouldn't mind coming back up here, and we won't have chairs, we'll just have the speakers stand with mics, and we can do a Q&A like in a kind of friendly, informal way. We're all colleagues here, and we can have Q&A at the end of each of the sessions. So save your questions for that. And I think that's it. We have an exciting day ahead. We will end with a very interesting panel discussion, which David Meeker will be leading. Thank you. And so hopefully this will actually allow us to have a lot of interesting conversations during the symposium, during the breaks, bring the community together, and hopefully make progress, as much progress as possible all together. So it's my honor to open the day today. Thank you all for coming. And let me now just skip to introducing our first keynote speaker to really open the day with some inspiring science and a really, truly inspiring story, I will say, behind the science, which is really remarkable. So our first keynote speaker is Professor Jonathan Weissman. Jonathan is an investigator of the Howard Hughes Medical Institute. He's a member of the Whitehead Institute right next door, and he's a professor of biology at MIT. His lab is really known for incredible breadth and depth of work, both in the way proteins fold and how things go awry in disease, which is an area that we share in terms of scientific interest, but also further, his lab really has done incredible innovative work in areas like ribosome profiling, CRISPR-IA screening in very innovative ways, including ways in which one edits, comes up with epigenetic editors and how those may be now deployed in different areas for therapeutics. And so I think we're here to hear an incredible story about that. And so without further ado, please welcome Jonathan Weissman.
Thank you so much. I have a feeling you can't hear me. Thank you so much for the kind introduction and, of course, for the invitation to speak today about our work on silencing prion disease causing protein. So back in sort of year zero of CRISPR, to the summer of 2012, right after Jennifer and Emmanuel had published their description of a single protein and guide RNA that could be a programmable nuclease, a few of us, Jennifer included, thought that we should take this beautiful nuclease and break its scissors so that it couldn't cut anything. And you might think, why would you want to do that? And the reason is now you had a programmable DNA binding protein. And if you then fused effector domains, so for example, in the very early work, we fused transcriptional inhibitors or transcriptional activators, you could now, in a programmable way, bring in epigenetic marks. In essence, we got a volume switch that allowed us to control the expression of any gene or any combination of genes. And if you are interested, as my lab had been for some time, in the function of different genes and in trying to understand what is information encoded in our genomes and what are modifiers of disease states, these has turned out to be extremely powerful tools. But from a therapeutic point of view, they weren't particularly useful because they were intrinsically unstable. As soon as you stopped expressing the Cas9, the epigenetic marks would dissipate often within a few days, and gene expression would go back to its normal level. So this motivated us to, instead of doing epigenetic marks on histones, for example, to bring in DNA methyltransferases that would, again, in a programmable way, be able to methylate the DNA. And the reason why this was transformative or potentially transformative is there are systems in your cells that once the DNA is methylated, that both ensure that represses transcription, but critically for propagating this. So now you could, in a hit-and-run way, bring in your epigenetic editor, your DNA methyltransferase, and permanently silence a gene. And the reason why, one of the reasons why these are so stable is that these are systems that are used by the cell to silence elements like parasitic elements like endogenous retroviruses. So that you're piggybacking on the cell's system for once you put these methylation marks in CPGs for propagating them. So for example, when DNA replicates, of course, only one of the two strands will inherit the methylation on the C. And of course, CPG is a palindrome, so if you have it on one strand, you have it on the other. So you then end up with two hemimethylated double helices. And there's then a so-called maintenance methyltransferase, DNMT1, that recognizes these hemimethylated states and then restores the methylation mark to the second strand. So, sort of to make a long story short, working off of beautiful work from Angelo Lombardo and Luigi Naldini, we brought together in a single effector protein that contained crab domain, 3L, which is a helper protein, and 3A, which is the actual methyltransferase, CRISPR-off machinery. And here is an example of this where you see there's transient expression over the course of a week or so, but the methylation and the silencing of the gene that's been targeted after a few iterations, we were able to get it extremely stable. So here's an example where James Nunes, who was a postdoc who led this work and is now assistant professor at Berkeley, silenced a gene through this transient expression of CRISPR-off, and then propagated the cells, in this case, for 15 months, and the vast majority, and made individual clones, and the vast majority of these clones remained completely silenced. And unlike the volume switch where you could tune down and up, the epigenetic silencing was really an off switch, like a sort of a digital, was it on or was it off? And the off was, as far as we can tell, completely unexpressed. And in work that was not ours at all, we work from Chroma Medicine, which, in full disclosure, I was a founder of, has used this in LNP delivery to the liver and shown that you can, with a single dose, and actually a very low dose, get permanent silencing, and this is in NHP data, permanent silencing of the PCSK9 gene, and this causes a dramatic decrease in serum cholesterol, LDL levels. So this sort of a proof of principle that, at least in a non-human primate, that these could be a therapeutic. And we thought there would be sort of the separation of church and state. We would use the epigenetic editors. We were scientists and about discovery. We would use epigenetic editors to understand how these marks are initiated and propagated and the basic biology of epigenetics. And chromas, or the chromas of the world, would use these for therapeutics. And that equation changed when one day this couple, Sonia Valib and Eric Minikal, came into my office and said, we're writing a grant that the goal is to fund up into an IND-enabling study for prion disease. And I knew Sonia and Eric. I'd interviewed them a decade earlier when they were going to graduate school, before they had graduated, and now we're running a lab at the Broad to treat and understand prion disease and prevent prion diseases. And I also, of course, knew Sonia's story, which is she was a Harvard-trained lawyer. Eric was a city planner. And at some point early in their career, Sonia's mother died of fatal familial insomnia, which is one of these inherited prion diseases, and that she herself had inherited this allele, and it was an allele that had very high penetrance. So she was, at some point, almost certainly, in her typically around 50s or 60s, was going to have the same fate as her mother. Unless, of course, they could do something about it. So they did, being Sonia and Eric, they were full in. They quit their day jobs, went to graduate school, learned science, and now we're running a lab. So with that, as you know, when it's not often you have the privilege of being asked to participate in something like this. So of course, we were all in on this. And so I should just say, the prion disease is one of the more broad, I think, motivations for using this as sort of a first proof of principle for treating neurodegenerative diseases, is that biology was so clear. And so I'll remind you, prions are infectious proteins where an endogenous protein, the prion protein, has misfolded into self-propagating conformation. Essentially, that acts as a seed. And when either through because of a mutation or spontaneously or through infection, you have these seeds. They then co-opt the normal protein and propagate and grow exponentially. And the other key piece is that we seem to be able to live, mammals, well without this prion protein. So you could knock out the prion protein, and if you weren't expressing the endogenous protein, you would have no substrate for this seed to act on. And we knew this from beautiful work 20 years earlier from Giovanni Malucci, who made a genetically engineered mouse with a Cre-Lox system where they could, in a defined time, knock out the prion protein across the central nervous system. And what Giovanni showed was that even after infection, if you prevented expression of new prion protein, you could block the prion disease. So silencing, we had a very clear biological hypothesis. Silencing prion protein will have a therapeutic benefit. The second, and I say this is work led by Edwin Newman, a graduate student in the lab, and Tessa Bertozzi, a postdoc in the lab. They quickly showed that the human prion and mouse prion protein had a promoter that could be readily methylated. So you transiently express the CRISPR off, and it caused permanent silencing of the prion gene, and that silencing was caused by methylation of the so-called CPG island upstream of the prion promoter. So now it was essentially a delivery problem. We knew that silencing of the prion protein from the engineered mice, that within the central nervous system would be sufficient to block the disease, even in an actively infected individual. And but we also knew that it could affect most of the CNS. So you really had to have a way of delivering across the CNS. And really the only approach at the moment to do that in an adult was through AAV. And fortunately, we have a wonderful collaborator, Ben Deverman, who is the world expert on AAV delivery to the CNS. The problem is that CRISPR, Cas9s are big proteins, and AAVs have small payloads. So just to give you an idea of the scale, the Cas9 alone was pretty much too big to fit in the AAV, let alone all the other bells and whistles. So this motivated us to then go back to an earlier technology, zinc finger proteins, which are programmable DNA binding proteins. They don't have the beauty of having the simple rules of a guide RNA, but through a lot of work, and work that was the foundation work for this work was done across the street in Carl Pebo's lab, we had rules that through a lot of engineering, you could, for a given site, design a zinc finger. And there's a couple of critical advantages of zinc fingers. One is that they're very small, so it's even a 6-finger zinc finger is only about 540 nucleotides, so they could readily fit in. In fact, we can put in 3 different zinc finger silencers in an AAV. The other is that they were human in origin. And the third is that these crab domains, one of the key components in the endogenous system for silencing these endogenous retroviruses, are almost always fused to zinc fingers. So it was really the natural way of delivering onto DNA the epigenetic silencers. And unlike the beautiful editors from David Liu, like base editor or prime editor, which really rely on idiosyncrasies of Cas9, the ability to do nick one strand and not the other, the ability to create a bubble that's a substrate for deaminase, we didn't need to do any of this. In fact, having a small DNA binding protein that would be right on the DNA and bringing the DNA methyltransferases was both more effective and more an elegant way of delivering their silencers. The problem we had, so this all seemed very simple. The problem is we made our zinc finger off, the equivalent of the CRISPR off, and even in vitro in cells, these were toxic. So why? And we showed that it was the DNMT3A, the methyltransferase, that was causing the toxicity. So why were these toxic? And bear with me, I'm going to have to tell you, go into a little bit of the nuts and bolts about DNMT3A to show how we solve the problem. So DNMT3A is actually a more complicated multi-domain protein, and it exists in an auto-inhibited form. What we and others have done is take only the methyltransferase, so removing this inhibition so that we circumvent this. The problem is you now essentially have a mutagen. You're overexpressing this uncontrolled methyltransferase that whenever it landed or wherever it was brought to inadvertently on DNA would cause these marks on the DNA. And because you had the DNMT1, the maintenance methyltransferase, once you put the marks, they could propagate indefinitely. And we also knew from the beautiful work that the way this autoinhibition is released, or one way, is through the histone tail. So basically, methyltransferase is sniffing around, and if it's a K4 methylation that is an indication of active transcription, the methyltransferase can't be activated. But if you have the K4 unmodified, it binds to this ADD domain, releasing the inhibition and allowing activation of methyltransferase. So Edwin had this idea that we could take the 3L and not express any methyltransferase or any enzyme at all in the cell and just recruit the endogenous methyltransferase. It wouldn't be in an active form, but it could recruit it through the 3L. But then he thought, what if we put on this histone tail in a way and played with the linkers in a way that it could bind to the ADD domain? This should, in principle, allow you to recruit and activate the endogenous methyltransferase. To make a long story short, this worked like a charm. That's our name. Coupled histone tail for autoinhibition release of methyltransferase, or CHARM. And through a lot of optimization, he was able to get this CRISPR CHARM to work in general, actually better than the original CRISPR off. So now, and he also showed that this eliminated, or largely eliminated, any of the toxicity they saw. So now we had a non-toxic zinc finger CHARM that could readily fit into an AAV, and we were now able to do the experiment with Ben's AAVs that could go reach across the central nervous system in a mouse. And really beautiful, almost in this first experiment, we got dramatic and brain-wide, and we showed it was actually in the neurons themselves, silencing of the prion protein to, in this case, more than 80% silencing, almost to the limit of detection. And this silencing was being mediated by methylation. So we had a reason to believe that it would be not transient, but permanent in nature. Because it was permanent, once you methylated the prion promoter, which we knew happened relatively quickly, continually expressing the CHARM could only cause off-target effects because you've done the on-target. So what we did in the work that was led by Tessa Bertozzi is she developed a self-silencing CHARM. So by tuning, she put in a binding site for the ZFP CHARM in the promoter that was expressing the CHARM itself, and then tuned this promoter, this binding site, such that the ZFP CHARM first silenced the endogenous gene and then more slowly silenced itself. So now you sort of have the perfect hit and run. It came in, silenced the prion protein, silenced itself, and you were left in the neuron with a silenced prion gene and a neutral piece of DNA that can't be expressed. And this worked, a lot of tinkering. We now have data actually out to 12 months. I'll show you here 6 months data showing that the CHARM, or the self-silencing CHARM, has very robust stability. But then we could look at what were the off-target effects. And what we saw was if we had continued expression of CHARM, prion protein was by far the biggest gene that was most affected, but there were clearly off-target effects on this. Whereas the self-silencing CHARM was really squeaky clean. So this one gene here was keratin, which means someone managed to probably contaminate it with RNA from their fingers or from the sample. But other than that, there was nothing above the detection limit. And the knockdown of the prion was actually, if anything, more effective. So now we were able to do the actual validation experiment in and I should say all of this stuff we were able to move so quickly because the beautiful system Sonia and Eric had set up that allowed us to quickly go into mice and test, go from cells to mice in a matter of weeks. So here you inoculate a mouse with the prion, wait till 120 days or so when the animal is actually now visibly sick and would die within tens of days after if you didn't intervene. Come in with an AAV, and you can see from these both the self-silencing or the non-self-silencing form are really able to dramatically increase the life of the animal. And I say this is, it's not a perfect cure in this, but it's also a very, very challenging problem. You're talking about an already sick animal that has a very high burden of the prion disease, infectious prion. So next step was all mouse. So we then went to making zinc fingers against the human prion using a humanized mouse model. There were some zinc fingers against the prion promoter in literature. They were quite effective when we made the ZFP charm at silencing, but they're really quite dirty. So through a lot of optimization, Edwin was able to turn this into this, and not only was he able to get rid of essentially virtually all of the visible off-targets, and this is with continuous expression, not long after with the self-silencing, so we think it will be even cleaner, but not only get rid of the off-targets, but actually have better efficacy against the prion. This was still a great silencer. And when we went into humanized mouse, we can get very robust brain-wide silencing, almost to the limit of detection. So what's the next step? We is really led by Sonia and Eric, have had an FDA Interact in February. They were very enthusiastic about the approach and really gave a lot of support for the strategy of using the mouse data and then going into human. So we're doing lead optimization, finalizing the promoters, the UTR, doing self-silencing and humanization. They have a contract CRO, Verogen, that's going to produce enough AAV for about 10 doses over the next year or so. GLP tox studies next year, and aiming either in second half of 2026 or early in 2027 for the IND study in symptomatic patients. So finally, where is it going to go from here? As I said, we're focused, of course, was on the prion disease. That is our central goal, but we also think that both by the AAV and de-risking all of this, and as a proof of principle, this could have much broader applications because this theme of protein aggregation and neurodegeneration is common throughout many inherited or spontaneous neurodegenerative diseases, including the triplet repeat diseases like Huntington's and even much more common ones like Alzheimer's disease. And so we hope that the ability to shut off specific genes in the brain could prevent these aggregates and potentially impact diseases that affect millions of people where there currently are minimal effective treatments. So with that, I'll end and thank everyone again. I mentioned most of the people along the way, but I think it's worth a shout out again for Edwin and Tessa and our wonderful collaborators, Ben Deverman, Sonia Valab, Eric Menikal, and also Fiona and Elisa, who just did a huge amount of work behind the scenes to make all this possible. With that, I'll end. Thank you.
Yes, Liz. We have a mic.
Oh, sorry. Thank you. I'm sure you get this question a lot, Jonathan. First, it's just incredible work, and I love seeing every time you present it. It's just so exciting. How do you think about, from the mouse experiments, the effects of dosing pre-symptoms versus post-symptoms? And then how do you think about that in the context of clinical trials where you already have quite a small patient population and you may want to dose them like when they're pre-symptomatic?
Yeah, I mean, so this is so the FDA was okay with the strategy both for symptomatic and pre-symptomatic, and it's a very different situation. So for people who are symptomatic, it's about 600 people in the U.S. are diagnosed with this a year, so 2 a day. So not a challenge to get patients, at least at this scale, because they're highly motivated. But it's 5 months, so we only had 2 months with the mouse. We have 5 months from onset of symptoms to typical time to death. And there's nothing you can do to intervene. So there's a very compelling case for going in for people who are symptomatic. I think that will be the first. But the hope is we're building in self-silencing from the very beginning. The hope is that will also that same vector could be used for pre-symptomatic. That said, we're like, the one thing from my voyeuristic views of pharma to understand is you always have a backup, and we're building in a V2 version that has some extra bells and whistles that so we're completely de-immunizing. So any peptides that could be presented by the MHC, we're putting in mutations. We're doing things that will make it even better zinc fingers, ultra specific, and better tuning of the self-silencing. So I think it's going to be, it's going to see what happens when we get into the clinic. We'll be able to, you can see prion levels from the CSF, so you can measure, have some measure of efficacy, and of course you'll have also a view of any toxicity caused by the AAVs. And at that point, it's going to be, as they say, a radio call about whether you go pre-symptomatic or you wait for a V2.